So this is chapter 12, which comes under plant physiology, which is respiration in plant. So since we have already done with breathing and exchange of gases in case of humans, so I thought we can switch over to this chapter and see how exactly oxygen is used. Did you go through the breathing and exchange of gases chapter? Do you have any doubts? Can you manage MCQs, everything? Okay, fine. Now listen, see, the chapter here is the respiration in plant. So exactly what does it deal with? So you have this equation. What is the equation? Food giving rise to energy. So energy will always be in the form of adenosine triphosphate, right, which is the energy currency of the cell. So how is this food, okay, is getting liberated as energy? So that is what is going to be dealt in this chapter. So which is respiration, which is also otherwise referred as internal respiration. Okay, internal respiration, otherwise referred as, what is the other name of it? Cellular. Since it is going to happen inside the cell, it is also called as cellular respiration. You get this point? Internal or cellular respiration, right? Okay, clear, Arka? So whatever we are going to discuss here, I said this is going to be a series of chemical reaction from FTE. Whatever we are going to discuss here, it is not just restricted to plant, but also it is going to happen including humans. So as for example, if you go to your first year MBBS in biochemistry, you will be, whatever we are going to read now, we are going to read that with some extra details, right? Everything is going to same. And this is how we are going to relate ourselves with plant, saying that animals and plants have evolved from a common ancestor, because whatever you see as a biochemical reaction in plant is also equally present in animal. Clear, Arka? Right. And you know that there is going to be a presence of oxygen and CO2 is getting released. So this is the thing. So how this energy is getting liberated from food is what we are going to discuss here, right? Fine. Now the initial paragraph of this particular chapter is going to say whether plant breathe, right? So we have two things here. One is the external breathing, internal breathing, or external respiration or internal respiration. So what is your opinion, whether the plant needs an internal respiration? Yes, obviously any organism, if we need ATP, it should be obtained through internal respiration. So plant definitely requires internal respiration. But you know, for this O2 is needed, and CO2 which is generated here has to be sent out. Whatever carbon dioxide produced as a waste has to be sent out. So in that case, see, in humans, for us to consume oxygen, we have a separate, you know, the lungs respiratory system with which we breathe in and breathe out, right? So in case of plant, you don't see any specialized structure present there, you know, helping a plant in this situation. So what happens is, plant do need oxygen, right? Plant do need oxygen. It has to also remove CO2. But how plant manages that is, plant have got some pores like stomata, through which oxygen directly gets into the system and CO2 gets out of the system, right? So what is the summary of this? So this food getting liberated to energy is a universal phenomenon happening in all living organisms, including plant. So for which food is required, oxygen is required. Where does the food come from? Plant food, where does the food comes from? Sunlight. Through plants themselves, they produce food by trapping the energy from sunlight, okay, through photosynthesis, and some of the food prepared by the plant is utilized for the plant only, and some food is consumed by our herbivorous, carnivorous. We ourselves call ourselves as heterotrophs, right? So food is obtained from plant. O2 is obtained from atmosphere. The point is, there is no separate respiratory system, okay, in case of plant, whereas it tries to manage a direct exchange between the atmosphere and plant cells through stomata, lenticels, etc., etc. That is the opening present on the surface of the plant cell. Clear, Arka? So food, oxygen, rendum theva. If both are there, then plant will be able to internally respire for it to get the ATP, right? And food, plant itself prepare, right? And see, plant prepares food only leaves which has got the chloroplast. Whereas root or something, the food prepared in the chloroplast will be translocated through phloem to the non-photosynthesizing part, which it will able to use it for the energy production, right? So food is required. Herbivorous will consume from the plant, carnivorous will consume from the herbivore. So the food keeps on, you know, you 12th law or law padichiruppa, 10% law, something like that, right? Okay, so this is what, and O2 directly from the atmosphere. So this is the concept. So plant definitely respires, right? Internal respiration is, so that is what is given in the initial paragraphs, nothing else, right? Okay, so before we get on to this concept, see, I'm going to talk about something here, right? Listen. Yeah, so let me use this space, this particular space. See, you have, say, food getting liberated to energy in the form of ATP, right? Now this E will always be ATP. As I said, ATP is going to be the energy currency of the cell. Now this food, what could be the probable candidates for this food? The food, what could be the probable candidates? Carbohydrate. So what else you eat to get energy? Protein, fat. These are all the probable candidates. So among carbohydrates, which is the principal carbohydrate you see here? Glucose. The principal carbohydrate is glucose. Glucose is what you see here, right? So these probable candidates which can act as a food liberating energy, ATP, you call this as a respiratory substrates. Okay, you call this as a respiratory substrates. You get this point? He has meant what cellular respiration here, right? And he has said, see, these are all the respiratory compounds that are oxidized during this process. See, any compound, in order to liberate energy, ATP, it has to get oxidized. Oxidation, your chemistry can define oxidation in one or two ways, but here the way that we need here is LEO. What is LEO? Laws of oxidation. So in case, let's say loss of electron is oxidation. So in case if I am a compound, I am going to lose the electron, I am getting oxidized. If I am going to gain the electron, I am getting reduced. So loss of electron is oxidation, gain of electron is reduction. So you have LEO and GER. What is GER? Gain of electron is reduction. So any compound for that matter, if it has to liberate energy, it has to get oxidized by losing electron. Loss of electron, gain of electron is reduction. And our concept you should have it in mind, because throughout the process you will encounter lot of oxidation and lot of reduction. Electron losing reduction. Electron losing oxidation. Electron gaining is reduction. Right? Idu note paniko. So are you okay with the comfortable initial concepts, everything? Okay, right. Listen here. So of which, as I said, as I have mentioned here, the major respiratory substrate is going to be glucose. So what we will do, though protein, fat, everything can act as a respiratory substrate, majority of our discussion will involve only carbohydrate, that too in particular glucose. So let's keep F is equal to G. What is G? Glucose, right? And then see how things happens. What will be the fate of a fat into energy? Oxidation of fat into energy, or oxidation and purida, because if it has to liberate the energy, it has to get oxidized. So what is the fate of the protein getting oxidized to ATP? Everything we are not that concerned here, but when you go to MBBS, you will have a detailed discussion regarding it. So at this moment we are concerned only with glucose. Clear? Right. So listen dear, I'll just give a overall summary. You want to note down this? Yeah. So I want to give a overall summary how things happen. Then we will see. Yeah. Yeah. See, listen. Let's consider this as a cell, right? I'm trying to write it here. Let's consider this as a cell. So I'll put a box. Right? So I'm considering this as a cell. So what is the cell? What do you see inside the cell? There is a nucleus. So imagine, we are not bothered about nucleus here, and this is going to be the wall. In case of plant, except animal, in case of plant, fungi, bacteria, there is going to be a cell wall. Whereas in animal, there is no cell wall, but only the plasma membrane. You are aware of it, right? So centrally you have got a nucleus, and you call this as a cytoplasm. You call this as a cytoplasm, right? And there are couple of organelles there in the cytoplasm, of which I need only the mitochondria, right? I am just making this way, mitochondria, which is a double membrane structure, right? Double membrane structure, fine. So this is the mitochondria. So mitochondria, right? So a glucose, a glucose now enters into the cell for it to get oxidized, because oxidation is the principal chemical reaction behind getting energy liberated from glucose. You are getting this point? Right. Now this glucose, what is the number of carbon of glucose? What is the formula of glucose? C6H12O6. So this is basically a 6 carbon compound. Now this undergo a breakdown called as glycolysis. So here glyco means glucose, lysis means breakdown, glycolysis, and we get 2 molecule of pyruvic acid. Pa means pyruvic acid. People say it as pyruvate or pyruvic acid. So when this is going to be a 6 carbon, glucose is 6 carbon, what will be the number of carbon of pyruvic acid? So it should be 3 carbon. It should be 3 carbon compound. So you have 2 molecule of 3 carbon pyruvic acid. This is going to happen in cytoplasm under the banner called as glycolysis, right? Now listen, once the clear, so glycol. So once the pyruvic acid is formed, then the fate of pyruvic acid depends whether O2 is present or not present. When I say O2 is present, I call it as an aerobic. When O2 is absent, I call it as an anaerobic. Exactly. So let's consider mostly our condition is aerobic. So in presence of oxygen, what happens? This pyruvic acid all the way enters into the mitochondria. Right? Enters into the mitochondria. Now the pyruvic acid is converted to acetyl CoA. Let's discuss these things in detail. I'm just giving an overall summary here. Acetyl CoA. Then acetyl CoA joins a cycle called as Krebs cycle. Right? Everything is happening inside the mitochondria, Krebs cycle, which is going to generate some NADH, FADH like compound, right? And those are going to join electron transport chain, which is happening in the inner membrane of the mitochondria. So mitochondria has got 2 membrane, outer membrane, inner membrane. Have you heard the term cristae? Rather, the inner membrane of mitochondria is thrown into many foldings called as cristae. Right? The cristae, you have some electron transport chain systems are there, ETC. Okay, enna irukku? ETC. Solradhu puriyudha? O2 presence la pyruvic acid will directly get into mitochondria. So mitochondria, pyruvic acid will get converted to acetyl CoA. Acetyl CoA will join Krebs cycle. Krebs cycle will generate lot of NADH, FADH2. Just what is that? Understand this, right? For the further now in-depth understanding, we will discuss these things in detail, right? So what happens is NADH and FADH2, okay, two things. So one is NADH, other one is FADH2. What happens? These two will join the electron transport system which is happening in the cristae, okay, which is happening in the cristae where ATP will be generated. So this is the overall process of aerobic respiration, which normally is at present happening inside us. If there is going to be even a fraction of second, in which these reactions are disturbed or paused, our life itself will be in a question mark. Life cannot happen. The suicide bombers, they used to have something called a cyanide. Once they are caught, they used to consume that cyanide chip in that or somewhere they used to have. So that is going to disturb this process. So that within a fraction of a second they lose their life. They are dead. Such an important, at present, a chemical reaction. So this is going to happen in presence of oxygen. So oxygen, aerobic, right? In absence of oxygen, right, this pyruvic acid in cytoplasm only, pyruvic acid can become either lactic acid or ethanol. Lactic acid or ethanol. So pyruvic acid is 3 carbon. So it can be either lactic acid 3 carbon, so 3 balance iodine, or it can be ethanol. That is either lactic acid or ethanol, not both. So what is the number of carbon in ethanol? 2. So 1, see, inga 3 irukku, inga 2 irukku. So 1 will be compensated with carbon dioxide. So this is what is called as fermentation. Okay, so this is called as a lactic acid fermentation. Okay, lactic acid fermentation, and this is called as alcoholic fermentation. ALCOHOLIC, alcoholic fermentation. So fermentation is one important concept that we would have gone through in the microbes in human welfare chapter in 12th, right? So this is the overall scheme of respiration. What happens during aerobic, what happens during anaerobic. So our next idea is we have to take each and everything like glycolysis, link reaction, Krebs cycle, electron transportation, each and everything, and see those things in detail. Detail in the sense, not too much of thing, but to an extent we will magnify these reactions and see what is there inside. Solradhu puriyudha, clear irukka? You want to note down this? You are okay with this? Right. Ipo adanga solran, breaking of the CC bond. So what is CC bond? Glucose irukna 6 carbon. I hope you know the structure of glucose. You would have read in your biomolecule chemistry. So once you understand the overall concept, you will be able to get these lines, okay, very comfortably, right? So of which, so next thing that we are supposed to do is, glycolysis. So this is the case. Glycolysis, clear arka? Glycolysis. See, this scheme of glyco here means, as I said, glucose, lysis means breakdown, right? There is a breakdown of glucose which is given by, the scheme is given by Gustav Embden, Otto Meyerhoff and J. Parnas. That's the reason why glycolysis is called as EMP pathway. Right? It is called as EMP. Glycolysis ke innuru per enna EMP pathway, because obviously the due credits has to be given to the people who have identified this, discovered this scheme, right? Glycose, sugar, right? Okay, fine. Now it is going to happen in cytoplasm of the cell, everything, right? So before this, you need to know the basic scheme of the biochemical reaction. So in chemistry, we have A chemical reaction, say A plus B give rise to C plus D. Yes or no? Now, such a chemical reaction happening inside a cell is what is called as a biochemical reaction. A chemistry happening inside a cell, biochemistry. So you call this as a, in chemistry, you call this as a reactant. Okay, you are going to call this as a product, okay, in presence of a catalyst. Whereas in biochemistry, this reactant will get the name substrate. In biochemistry, you won't call it as a reactant, but you will call it as a substrate. The product is, the name is as it is product, but instead of catalyst, you see a biocatalyst, right? You see a biocatalyst, which is otherwise referred as, what is it? Enzymes. So, biocatalyst is an enzyme. And to my understanding, catalyst just speed up the reaction in case of chemistry, but enzymes not only speed up the reaction, in absence of enzyme, reaction cannot happen. This is my understanding. I may be wrong. Okay. When catalyst A plus B will get somehow transformed to C plus D, but it may take some more time. But whereas, if you are going to remove an enzyme, A plus B remain as A plus B. So, reaction happens mostly only in presence of enzyme, though there might be some exceptional cases, but generally this is the situation. Clear arka? Idu purida? Ipo A to B. This is one chemical reaction. A is the substrate 1, B is the product 1 in presence of enzyme 1. This is a biochemical reaction. Now, this B will become the substrate 2. As I said, you are starting from here, you are arriving at university, so origin, destination, further you have to go to Chennai. So, your destination 1 will be the starting 2. Then probably you land up at Kalpakam. So, substrate 2. You get C. This is going to be product 2, which is substrate 3. So, this is one reaction, this is one reaction, this is one reaction. You see lot of chemical reactions getting linked like a chain, and you call this as a biochemical pathway, right? You call this as a biochemical pathway. Now, your glycolysis is an example of a biochemical pathway. அதாவது ஒரு 10 ரியாக்ஷனை சேர்த்து நீங்க இது பண்ற சொல்றது புரியுதா? Sometimes what happens, this biochemical pathway, the final product will join with the initial substrate. அப்படியே போயிட்டு ரவுண்டா வந்து இப்போ நீங்க சென்னையில இருந்து you go to Pondicherry, I mean சென்னையில இருந்து Pondicherry, you come via Dindivanam, then you take via ECR, அப்படியே வந்து again you go to Chennai. So your final destination and initial point both are same. That connect ring mari connected. So you are able to get, then the biochemical pathway will be referred as a biochemical cycle. So both are same. Biochemical pathway, biochemical cycle, both are same. But biochemical pathway, both have got some linked series of reaction. But in biochemical pathway, the first point and last point both are different. There is no connection. Whereas in biochemical cycle, both are connected. எல்லாத்துலயுமே இதுதான். Substrate 1, product 1 will behave as the substrate 2 for the second reaction. So you keep seeing these reactions getting connected to each other. Clear arka? So with this, let's start with our glycolysis. So இதுதான் நமக்கு கொடுத்திருக்கிற glycolysis, right? Glycolysis, right? So though the enzyme name has not been mentioned, but we are supposed to know சில enzymes பேர்லாம் நமக்கு தெரிஞ்சிருக்கும். இது வரைக்கும் you are able to understand till now, right? Okay, right. Listen. So let's start with glucose. So where does this reaction happens, dear? Right. Glucose is a carbohydrate. Where does the reaction happens? In which part of the cell? அதான் உன எழுதிக்கோன்னு சொன்னேன். Which part of the cell? Cytoplasm. So what is the final product of your glycolysis? What is the final product? You have pyruvic acid. Pyruvic acid will enter mitochondria in presence of oxygen. நான் சொல்றது புரியுதா உனக்கு? Right. So this is and you'll also note that this is going to be the common pathway for both aerobic and anaerobic. Pyruvic acid is what is going to get further converted into lactic acid or ethanol depending upon the enzymes present. So this is a common pathway. Okay, from pyruvic acid only the fate will be different depending upon the presence and absence of ஆக்சிஜன். நான் சொல்றது புரியுதா, கிளியரா இருக்கா? ரைட். இப்போ நான் சொல்றதெல்லாம் கவனிச்சேன், நோட் டவுன். இப்போ நான் சொல்றதெல்லாம் if you are going to understand, then whatever is there here will be easy for you, right? So let's start with glucose, right? So first reaction, glucose 6 carbon is getting converted to glucose 6 phosphate. So what do you mean by the term glucose 6 phosphate? So you know what is glucose. What do you mean by the term glucose 6 phosphate? Addition of? No, it is going to be apdina, it will be called as a hexaphosphate. So the 6 indicates the carbon to which a phosphate is added. So this can be called as a glucose monophosphate. So 6 indicates the carbon number. See, glucose is going to have 6 carbon. So you have ATP here. What is ATP? Adenosine triphosphate. What is ADP? Adenosine? Adenosine diphosphate. So what is the difference between ATP and ADP? How many phosphate difference? 1. Appo you have to subtract 1 phosphate from ATP for you to get ADP, and that 1 phosphate will get added to 6th carbon of glucose, getting glucose 6 phosphate. நான் சொல்றது புரியுதா, கிளியரா இருக்கா? இதுக்கும் ஒரு phosphate வித்தியாசம் இருக்கு. 6ங்கிறது number of position of carbon, right? The enzyme is going to be உன் bookல எழுதிக்கலாம். Enzyme is going to be hexokinase. Enzymes are some enzymes are important. Hexokinase. Is that clear? Right. Now glucose phosphate to fructose 6 phosphate. So what are glucose and fructose? Isomers, functional isomerism, right? So both are same. So you see an enzyme phosphoglucoisomerase. You get this point? Purida? Right. Now you see, based on the knowledge you got here, can you say what is happening in this reaction? See, this is the product 1, which is the substrate of the reaction 2, and this is the product 3, which is the substrate of the reaction 4. That's how the reaction is getting linked here. What is this fructose 1,6 bisphosphate to fructose 6 phosphate? What difference do you see here? At which car? Already there is a 6. இங்க 6 இருக்கு, இங்க 6 இருக்கு. இங்க என்ன எக்ஸ்ட்ரா திங்க் பண்ணு, எக்ஸ்ட்ராவா என்ன இருக்கீங்க? அடே, what is this? What is this? What is this? 1, இருக்கா இல்லையா? Fructose 1,6 bisphosphate. So what do you mean with the term bis? 2. So already where the phosphate was there, now where it has got added? கவுண்ட் பண்ணி வாழைப்பழம் ஜோக் மாதிரி சொல்றேன். என்னடா அங்க 1 இருக்கே, அந்த 1 எதுக்குடா இருக்குங்க? ஃபர்ஸ்ட் பொசிஷன் and 6th பொசிஷன். அப்ப ஏற்கனவே வேர் தி பாஸ்பேட் வேற எங்க இருக்கு? 6thல. புதுசா எங்க ஆட் ஆயிருக்கு? புரியுதா? ஏன் இதுல என்ன இவ்ளோ இதுக்கு 1 இங்க 1 இல்ல, இங்க 1 இருக்கு. Bisphosphate, extra phosphate is getting added, and where do you get the extra phosphate from? ATP, ADP. This is going to lose 1 phosphate, which is getting added to the first carbon. Thereby this become fructose 1,6 bisphosphate. Purida? The enzyme is important enzyme, phosphofructokinase. Enzyme phosphofructokinase. Clear arka? Now listen. Now until then 6 carbon maintain out. From glucose to this point 6 carbon is getting maintained. Now here what the split is going to happen? Now 1 fructose 1,6 bisphosphate, 1 will become triose phosphate, அதாவது glyceraldehyde 3 phosphate. இது ஒரு 3 carbon compound. Another is dihydroxyacetone phosphate. Triose na 3 carbon னு அர்த்தம். Triose na 3 carbon. So this is going to get split into 2 3 carbon. One is DHAP, இங்க கொடுத்திருக்காங்க பாரு 3 carbon. Other one is glyceraldehyde 3-phosphate. See, this dihydroxyacetone phosphate in turn becomes glyceraldehyde 3-phosphate. இதுக்கு தனி ஃபேட் கிடையாது. இங்க ரியாக்ஷன் டபுள் வே ரியாக்ஷன் போட்டுருக்கு பாரு, ரிவர்சிபிள் ரியாக்ஷன். ஸோ இதுவுமே என்ன ஆகிடும்? அப்போ how many glyceraldehyde 3-phosphate you will get with from one glucose? How many G3P? ஒரு குளுக்கோஸ்ல இருந்தா நீ பார்த்துன்னு வர. There is one G3P here and DHAP also become G3P. So how many G3P in total? 2 எப்படி 6 எப்படி வரும்? 2 மொத்தமா சேர்த்தா 6 கார்பன், each 3 கார்பன். அப்போ எனக்கு 2 molecule of G3P will be there. கிளியரா இருக்கா? நான் சொல்றது புரியுதா? கவனி. அடுத்தது இது வரைக்கும் கிளியரா இருக்கா? அடுத்தது இந்த glyceraldehyde 3-phosphate இருக்குல்ல, now this கவனி, glyceraldehyde 3-phosphate loses electron. So when I say it is going to lose electron, what sort of chemical reaction that is going to undergo? Oxidation. So this is getting oxidized to, okay, இங்க எல்லாமே 2 னு கொடுத்திருக்கான் பார், into 2 ஏன் ஒன்னு இங்க இருந்து வருது, ஒன்னு ஆல்ரெடி ஃபார்ம் G3P, other one obtained from DHAP, right? அப்ப என்ன ஆகும்? 1,3 bisphosphoglyceric or glycerate அப்படின்னு சொல்லலாம். 1,3 bisphosphoglycerate. புரியுதா நான் சொல்றது? தெளிவா இருக்கா? கவனி. இப்போ இது எலக்ட்ரானை லூஸ் பண்ணுது. எலக்ட்ரான் லூஸ் பண்ணும்போது you have a molecule here called as NAD+, nicotine adenine dihydrogen something, it has got some expansion name. Now this NAD+ will accept the electron from glyceraldehyde 3-phosphate and it is getting reduced to NADH. அப்போ what is happening here? Oxidation or reduction? எங்க oxidation நடக்குது? where oxidation is happening? what is oxidation? ஆனா லியோ கெர்ன்னு சொல்லிட்டேன், அப்புறம் நீ வந்து லியோனா என்ன? what is Leo? electron is oak என்ன? oxidation. அப்ப நீ gain of electron சொல்ற. லியோ கெர் glyceraldehyde 3-phosphate என்ன பண்ணுது? எலக்ட்ரானை லூஸ் பண்ணுது. அப்போ what is happening? It is losing. அப்போ when I lose the electron, இப்போ நான் I see assume that I am glyceraldehyde 3-phosphate, I am losing the electron. நீ அக்செப்ட் பண்ற, அப்போ நான் என்ன ஆகிறேன்? I am losing the electron கண்ணா. அப்போ what is happening? What I am doing? What is happening with me? Today I am losing the electron. Loss of, நான் ஒரு கோட் வேர்டு கொடுக்குறேன், அப்போ நீ கன்ஃபியூஸ் ஆகிற. கோட் வேர்டு லியோ. what is Leo? yes, Lக்கு என்ன அர்த்தம்? loss. Eக்கு என்ன அர்த்தம்? electron. what is meaning of O? then I am losing electron. so what I am undergoing? oxidation. you are gaining electron. what you are undergoing? அதானே இங்க. அப்போ glyceraldehyde 3-phosphate is losing electron. so what it is undergoing? who is gaining the electron? NAD+. it is getting, after accepting electron, it is becoming NADH. so what is happening with NAD+? reduction. so what is happening? oxidation, reduction. which is happening in total? oxidation and reduction. so redox reaction is happening. புரியுதா? so இந்த மாதிரி oxidation reduction ரெண்டும் நடந்ததுன்னா அங்க ஒரு enzyme G3P dehydrogenase. the dehydrogenaseங்கிற enzyme, it has got an other name, oxidoreductase. clear இருக்கா? it has got other name, oxido. so wherever you see NAD+ or FAD+, there is a reduction happening. NAD+ to NADH, FAD+ to FADH2. it is accepting electron. so who is giving the electron? somewhere here. அப்போ உனக்கு எங்கெங்கெல்லாம் இதை நீ பார்க்குறியோ, wherever you see this presence of this, it means that there is a redox reaction. I'm not sure whether I can call it as a redox, but I can say both oxidation reduction reactions are happening. அப்போ அங்க enzyme கண்டிப்பா என்னவா இருக்கணும்? oxidoreductase. நான் சொல்றது நல்லா புரிஞ்சுக்கோ. what is the other name of oxidoreductase? dehydrogenase. புரியுதா? கிளியரா இருக்கா? மறக்க மாட்டியே? ரைட். அடுத்தது இப்போ நோட் டவுன் சம்திங். கிளியரா இருக்கா? இப்போ அடுத்தது 1,3 bisphosphoglycerate, ரைட்? ஸோ இது 2 விட்டுட்டு 2ங்கிறது into 2, அதாவது 1 G3P coming from DHAP, ரைட்? ஸோ இந்த 2 விட்டுட்டு இப்போ 3-phosphoglyceric acid. அப்போ உனக்கு இதுக்கும் இந்த ரியாக்ஷனுக்கும் what difference do you see here?
This phosphate.
அப்போ இங்க how many phosphate are there? 2. இங்க which carbon the phosphate is there? First and third. இங்க அப்போ ஒரு phosphate loss இருக்கா? அது ADP கூட combine ஆகி it becomes ATP. clear இருக்கா? right? அடுத்தது 3-phosphoglyceric acid becoming 2-phospho. So what is happening here in this? What is happening? 3-phospho to 2-phospho. பாரு எப்படி reduce-னா என்ன? What is reduction? இங்க where is gain of electron happening? இப்போ என்ன what is happening? This is 3-phosphoglyceric acid. This is 2-phosphoglyceric acid. So என்ன change இருக்காங்க அதை பாரு. One phosphate which is sitting at the third carbon has got shifted to second carbon. Reduction சொன்னாலே NAD plus ஓ FAD plus ஓ ஒன்று வரணும். அது இல்லாமல் ரிடக்ஷன் சொல்லாது. அதுதான் எலக்ட்ரான் அக்செப்ட் பண்ணும். அப்படி லியோகர் வச்சு ஞாபகம் வச்சுக்கலாம். இல்லை காசு கொடுத்தா உயர்ந்துருவாங்க. ஒரு பேச்சுக்கு சொல்கிறேன். காசு வாங்கினா ரெடியூஸ் ஆகிடும், குறைஞ்சி போயிடுவோம், சம்திங் அந்த மாதிரி. யூ ஷுட் ஹேவ் சம் டாக்டிக்ஸ் டு ரிமெம்பர் தீஸ் திங்ஸ், ரைட்? ஓகே, அடுத்தது 2-phosphoglycerate become phosphoenol pyruvate. The enol is some sort of chemical linkage or something, right? அதுக்கப்புறம் இந்த phospho again it combines with ADP become ATP. So only pyruvate is remaining, right? இந்த phospho இங்க வந்துரும். இப்போ இதுக்கும் பார்த்தீங்கன்னா வெறும் pyruvic acid. Pyruvate தான் pyruvic acid. AT னா it is nothing but acid. 2-phosphoglycerate means 2-phosphoglyceric acid. clear இருக்கா? அப்போ என்ன ஆகும்? இங்க உள்ள அந்த phospho enol could be a linkage, right? Phospho pyruvate enol. Enol is the bond or linkage which gets broken. Phospho joins with ADP becomes ATP and this pyruvate becomes pyruvic acid, which is the final product of which one? Glycolysis happening from glucose. clear இருக்கா? The steps of glycolysis.